So the first speaker I'd like to introduce is another Dr. Neen Pyzocha, who just defended her PhD very recently. She'll be giving you an introduction to the basics of CRISPR.
So, good afternoon, everybody. My name is Neen Pyzocha, and as Winston just said, I just finished my PhD here in the Zhang lab, and I'm really excited to be kicking off Genome Engineering 5.0 with a talk on CRISPR Basics. So, the purpose of this talk is targeted to those of you who may be new to working with CRISPR Cast technologies. But if you've worked with CRISPR before, hopefully this will be a good reminder of some of the basics. So, let's begin. So here's what I plan on covering in this section today. First, I'll give a brief introduction into genome engineering, and then describe CRISPR systems. Next, I will give some examples of available tools for genome engineering, describe guide design, gene editing workflow, along with delivery, and then describe some assays to monitor editing. And then lastly, describe some additional application of some CRISPR-Cas tools, as well as touch a little bit on HDR. So first, an introduction to genome engineering. So genome editing utilizes processes present in mammalian cells to repair double-strand breaks. So the predominant pathway is NHEJ, as shown by this red arrow. It is an error-prone process that often leads to small insertions and deletions, also known as indels. And if this occurs within a protein-coding gene, it can lead to a frame-shift mutation and then an early premature stop codon. And so targeting a double-stranded break in a gene is a good way to knock out a gene. Another way to repair double-strand breaks is through HDR, or homology-directed repair, as long as you have a template. If cellular conditions are right and the template is present, a cell may repair the DNA using this precise genome gene editing method. So now that we know that double-strand breaks can lead to gene editing, how can we target a double-strand break to a region of interest? And we do this using programmable DNA nucleases. In years past, as shown here, we've done this using tools such as zinc fingers or talons, and these are functional and very effective. However, they rely on a targeting mechanism that's conferred by modular protein binding domains that are laborious and low throughput to engineer in the lab. The CRISPR-associated Cas proteins have recently been employed for targeting DNA editing because they are easy to use, precise, and specific. Cas proteins use a short guide RNA to target a protein to a specific nucleic acid sequence. So now that I've briefly introduced CRISPR, I'm going to now go into some of the specifics on how this works. CRISPR is an acronym standing for Clustered Regularly Interspaced Short Palindromic Repeats, and it is a naturally occurring bacterial immune system. CRISPR-Cas is composed of two main components, the CRISPR locus and the Cas genes. And the acronym CRISPR describes what the CRISPR locus looks like. It is a repeat sequence interspaced by spacer sequences, which are unique. So you have unique sequences and then these repeat sequences. Also are these Cas genes that are transcribed and then translated into proteins. The CRISPR locus undergoes transcription to produce a long pre-CRRNA with multiple spacer and repeat units. This pre-CRRNA is then processed into individual repeat spacer units. This is the functional unit for targeting. These then associate with Cas proteins and then can target nucleic acids. And as you can see, for example, this green one can target this green bacteriophage nucleic acid. CRISPR systems are actually quite diverse, and they come in many different flavors. It's not just a single CRISPR-Cas system that occurs in different bacterial genomes. And there's quite a lot of nomenclature studies that have been done on them. And they're most broadly characterized into two major classes, class 1, and class 2. And the main differences between class 1 and class 2 is that class 1 requires multiple proteins to come together to form a complex to mediate interference, whereas class 2 relies on a single large effector protein to mediate interference. For simplicity, class 2 CRISPR systems are used for mammalian genome editing simply because they're simpler and only require one protein and one guide to target a nucleic acid. And so from here on out, all of the Cas proteins that will be described are going to be the class 2 large single effector Cas proteins. To describe some of the key components of CRISPR-Cas-based targeting, shown here is the commonly used Cas9 protein from the bacterium Streptococcus pyogenes, or SP-Cas9. The protein, Cas9 is shown here in yellow, specifically interacts with a guide RNA, which is composed of two different parts. The sgRNA scaffold, also sometimes known as the direct repeat, is shown in red and forms a unique secondary structure that is recognized by the protein. The guide RNA also has a second part known as a protospacer, and this protospacer forms Watson-Crick base pairs with the nucleic acid, in this case DNA. Another key component for Cas9 targeting to DNA is this sequence called the PAM. And the PAM stands for the protospacer adjacent motif and is required for Cas9 binding and subsequent cleavage. For SP-Cas9, the PAM is NGG, but this PAM sequence can vary depending on which ortholog or Cas9 protein you're working with from different bacterial species. Cas9 makes a blunt cut, as shown by these red arrows, a few bases upstream of the PAM. Now, one thing to note is that Cas9 can target either strand of DNA. And due to the antiparallel nature of DNA, it's important to note that the guide is always complementary or base pairs with the strand of DNA not containing the PAM. And so if you're targeting the bottom strand, or if you're targeting this strand down here, the PAM is always complementary to the non-PAM strand. So this is just an important thing to keep in mind when designing your experiments. So shown here is the crystal structure of Cas9 and its domain organization. Cas9 has two nuclease domains, RUVC and H&H, each which cleaves one strand of the double-stranded DNA molecule. It has these catalytic residues, and if these are mutated to alanine residues, a catalytically inactive form of Cas9, which is also known as dCas9, can be created. dCas9 can be used for programmable DNA binding, can be used as a programmable DNA binding protein, and is used in many binding applications, some of which will be described later. Finally, one other thing I'd like to discuss about Cas9 is its mechanism of binding and cleavage. So shown here is the Cas9 protein with its guide RNA. And the first way that it interacts with the DNA is that it recognizes the PAM, and it binds the PAM sequence. Upon finding a PAM sequence, it then begins to melt the DNA directly upstream of the PAM and forms pairing between the guide and the target. With extensive pairing, you get cleavage of the DNA. However, if you have partial pairing, this results in no cleavage. So one of the important reasons why I like to show this slide is that it gives you some insight into the binding and cleavage mechanism. And this can also give you some ideas on how certain mismatches may be tolerated. And that's key for understanding Cas9 specificity. So Cas9 binds relatively promiscuously throughout the genome, since it first relies on binding at the PAM sequence. And it only cuts when there's really extensive pairing and cleavage. And so this informs that mismatches are not tolerated, or much less tolerated at the PAM sequence, since that's the first sequence that Cas9 and the guide interacts with. It also is less likely to cut if there are mismatches on DNA bases proximal to the PAM. Lastly, if there are multiple bases, mismatches or indels, Cas9 is also less likely to cut. But Cas9 can cut at non-targeted sites if the mismatches occur at the distal ends from the PAM, or if there's a single mismatch or indel. And so again, these are important to keep in mind for specificity, and when choosing a guide, you can see that predicted off-target cleavage occurs at targets that seem to line up with some of these suggestions. So next, I'll go through some available tools for genome engineering. When working with CRISPR in your lab, the first thing to do is really to consider your project's needs. And every project will have different needs and requirements, and these can be very key to help you select which CRISPR-Cas system that you decide to use for your experiment. And so when considering your project's needs, one of the first things you can think about is what species are you working with? There are multiple CRISPR enzymes that have been optimized to work in certain species. Next, is it very important to have minimal off-target cutting? For example, if you're making a targeted knockout and you want it to be just in the gene of interest, then having low off-target may be very important for your experimental design. Next, if you're working with a particular cell type or in vivo in mice, let's say, a certain delivery method may be required to target your certain cell type of interest. And so therefore, knowing the delivery method may help you determine which reagents that you use. And then finally, some enzymes are better suited to target GC-rich regions, whereas others are better targeted for AT-rich regions. And so understanding your genomic locus that you're working with can help you better understand which tool may be ideal for you to use. So after you consider your project's needs, you can determine which CRISPR nuclease to use, and then computationally identify your targeting sequence, and then you can test it. So shown here is a page taken from AdGene, and AdGene is a really great resource, especially for academics, to acquire many different plasmids from many different labs. And you can really do many different things. You can cut DNA, you can activate, you can visualize, and so the number of tools that are available is actually really exciting, especially over the years, the number of plasmids deposited at AdGene are really, there's quite a lot of them. A quick note, so AdGene does have a table here in the lobby of Genome Engineering 5.0. And so if you have particular questions about which plasmids they carry, or if you want to chat with them, definitely recommend chatting with them. But for the sake of this basics talk, I'm going to focus solely on cutting CRISPR-Cas tools. So if you're looking to cut the DNA, even with this one particular application of CRISPR-Cas tools, there are many different options that you can choose from. So first off, you can choose many different orthologs of Cas9, orthologs meaning coming from different bacterial species. For example, two orthologs that may be interesting to use include SP-Cas9, which is commonly used, and then SA-Cas9 is another one. Again, if you're looking to deliver using certain delivery methods, for example, viral methods, they have lentivirus as well as AAV-specific backbones that are very important to use in those particular cases. Also, they have Cas9s driven by different promoters, different selectable markers. There are additionally some Cas enzymes that have engineered to be higher specificity as well, so therefore lower off-target cleavage. There are even some enzymes that have altered specificity, so have been engineered to have different PAM requirements. And then lastly, there are even some inducible Cas9 constructs. So enhanced specificity nucleases are very interesting, and so I'd like to briefly describe a few different methods to have enhanced specificity. The first is by using two Cas9 enzymes. You can either have paired Cas9 nicases, described by Rand et al., and in that, you nick the strand of DNA instead of having a double-strand break. And if you pair two nicks together, it can result in higher specificity. An additional way is by using dCas9s that have a fused FOC1 endonuclease. So this is kind of similar to working with talons, where the Cas9s themselves are catalytically inactive, but the FOC1 nucleases, when they dimerize, are able to cleave the DNA. And therefore, you need to have specific binding in two different sequences. If you're looking to work with a single Cas9 but want enhanced specificity compared to the wild-type Cas9, there are two different versions. There's ESP-Cas9 as well as SP-Cas9-HF1, which have been engineered to have higher specificity. And so those particular Cas9s may be of interest for you to use in the lab. I mentioned that there are multiple orthologs of Cas9 that can be used by researchers. Again, SP-Cas9 is one of the most commonly used ones. It's very robust, works very well, but it's a bit large compared to another ortholog known as SA-Cas9. One of the key interesting things about SA-Cas9 is that it's very small, and it's small enough to fit into an AAV viral backbone. And if you've worked with AAV before, you know that it has very small packaging limits. Besides SA-Cas9 and SP-Cas9, there are additional orthologs available, including N. meningitidis and S. thermophilus, but they have slightly longer PAM sequences than SP-Cas9, which is a very short PAM sequence, and is very easy to target throughout the genome. So up until now, I've been describing Cas9, but there is another single effector DNA nuclease that has been recently described, and it's called CPF1. So it is a completely different protein, but it also cleaves DNA. It has a RUV-C domain. And another interesting thing about CPF1 is that it's able to cleave its own CRISPR array. And so before moving on, here is a table describing some of the orthologs and so describing SP-Cas9, SA-Cas9, and CPF1, and some of their differences between them. So again, SP-Cas9 is very consistent, has a nice short PAM. SA-Cas9 has a longer PAM, but it fits into AAV. So this is very key to keep in mind. CPF1 has a T-rich PAM, and it also is able to cleave its own CRISPR array. And so these tools are available, and they're exciting to use. And I'd like to also briefly note that there have been other new proteins that have been discovered. So again, these are single effector Cas enzymes that can cleave nucleic acids, including Cas12b, Cas12c, Cas13a, Cas13b, Cas6, and Cas-Y. If you're interested in some of these new enzymes and what they do, I'm not going to go through them here, but some of these will be discussed in the Frontiers in Genome Editing talk here later today. So next, after deciding on which Cas enzyme you'd like to work with, it's then good to design your guide. And so here are some important considerations when you're designing your guide. So first, again, you need to figure out which Cas enzyme you're going to use, because this dictates your PAM sequence that needs to be adjacent to the target, the length of the guide, and the direct repeat sequence that's associated with that particular Cas enzyme. Again, you also need to know which species you're working with. And this is key because if you're working in a cell line or in an animal, this can dictate it can dictate the off-target analysis and where cleavage events may occur outside of your target area. And then lastly, when selecting your guide, it's important to keep in mind if you're doing a knockout or a knock-in, because this can slightly alter where a cut site may occur. A knockout needs to occur in an early constitutive exon, whereas a knock-in, so let's say if you're trying to knock in a large piece of DNA or do HDR, usually needs to be as close as possible to the site of alteration. And so the overall goal here is to select a target in your region of interest that satisfies the requirements, so for example, the PAM, and reduces the probability of off-target cleavage. There are many online tools that are available to help you select guides. And these are key to use, especially if you're looking for genome-wide off-targets, because genomes are very large, and these tools are very quick and easy to use. Here, with so many tools, one might ask, what's the difference? Why would you use one tool over another tool? And here I reference a nice review by Tycho et al. That goes through some of the differences between some of these different tools. Some tools support many different genomes. Some tools are better for looking at particular orthologs of Cas9. And then another key thing to note is that some tools consider off-targets with indels. So this was later found that certain indels can be tolerated by Cas9 and result in cleavage. And so if you're looking for limiting the number of off-target or having an idea of which off-targets are possible, selecting a tool that considers off-targets with indels may be very important for you to use. And so since this is a basics talk, I'd like to briefly go through an example. And just for today, I've decided to work with Guidebook. So Guidebook is by Desktop Genetics, and also like to briefly let you know that Desktop Genetics also has a table in the lobby. So if you have particular questions about this tool or anything else they do, I highly recommend that you seek out their table and ask them some good questions. So if you're using Guidebook here, to select a guide RNA, it'll first ask you, what are you using this guide RNA for? And so for today's example, I decided to create a guide for a knockout. When you select a knockout, it'll next ask which genome you're working with, again, to figure out the genome-wide off-targets, and then ask you which gene are you interested in knocking out. In this case, EMX1 is one of our favorite genes in the Zhang lab to knock out or to work with. If you're looking to simply make a cell line, this is actually a really interesting feature that Desktop Genetics will let you know if there's a cell line that's already commercially available and you can get. If you're looking to make a specific mutation or change, then you might want to make your own guides. And so after selecting your gene, you press Select Guides. Right, so you select genomic build and then gene of interest. So this is the page that comes up. It shows you multiple different isoforms. And then right here, these are all showing you where guides are located. For example, this is number 35, this is the PAM sequence, and this is the target. Here shows multiple different guides that could be used to target this particular gene. Here, you can filter based on predicted activity. And you can also select guides to complete off-target analysis, as shown over here. And the cool thing about this off-target analysis is that it gives you off-target hits or predicted regions within the genome that are expected, or it's possible that an off-target cleavage event may occur, as shown in this table right here. Another key thing to keep in mind is that this off-target analysis gives you an idea of where this off-target hit occurs. For this particular guide, you see that there is one potential off-target within a coding region, and so this could be potentially not good depending on the guide that you're using. Whereas if it targets a non-coding region, although this could alter non-coding areas of the genome, may be all right if there's a couple potential off-target hits in a non-coding region. So now that I've described how to select the Cast tool that you're working with and to select your guide, now I'd like to go through the workflow of how to edit your genome and deliver it. So here's a brief timeline to test a guide or to generate a knockout cell line. So first, during the first day, we'll require just in silico preparation. So choosing your Cas9 or choosing your Cas protein to work with, selecting your guide, and then ordering reagents. Next, you need to construct your reagents, so do some molecular cloning. And then you can transfect into a cell line if you're working with a cell line and test it. To see if your guide worked, you can assay a pool of cells for on and off-target cutting. If you're looking to simply create a knockout cell line, after you assay your pool of cells and you see that you have a knockout cell line that is generated, you can then do clonal expansion and then screen these clones to see which ones have your knockout of interest. So there are two main ways that you can transfect and test your guides, for example, using Cas9. You can first clone your guide directly into a plasmid that contains a promoter for the guide, often using the U6 promoter, and then also a promoter for Cas9. And doing this method, so putting the guide into your plasmid, often results in higher efficiency, because if the cell receives the plasmid, it gets both the guide and the enzyme. And it's a single construct of transfect. However, if you're working with multiple guides, you can transfect them individually. So you can simply create a PCR product that has the promoter as well as your guide RNA, separate from a plasmid that simply has a promoter driving Cas9. Again, this is easier if you're working with multiple guides because all you have to do is create multiple different PCR products, mix it with your plasmid, instead of cloning each one individually in. So briefly, a note on transfections. So the key thing here is that transfections will really depend on the cell type that you're working with. So the reagent that you use and the protocol that you use will depend on your own cell type. So it's best to reference the literature or ask other people who have worked with your cell line of interest. For our lab, and if you're just trying to test out a guide to see if it works, we often use HEC293 cells. You can also use N2A cells. And we use the transfection reagent lipofectamine 2000. And here's a brief description of our transfection protocol. We plate cells so that they're about 70% confluent the day of transfection. And then we transfect about 500 nanograms of the plasmid, and if it has the guide with it, or 400 nanograms of the plasmid with just Cas9, and then 100 nanograms of the PCR product. Again, this will really vary depending on which cell line that you're using, but just to give you an idea of how we've done it in our lab. There are many other delivery methods that you can use to deliver your Cas9s to test them or to use them in an experiment. So again, you can transfect using these mammalian expression vectors. If you're doing lentiviral transduction, you need to have a lenti-based backbone. You can also have other selectable markers. If you're doing AAV transduction, if you want to include the Cas9 and the guide on the same plasmid, it's ideal to work with saCas9 because of a small packaging limit. Also, you can create Cas9 mRNA as well as the guide RNA in RNA form and microinject or electroporate into your cell line of interest. Lastly, some people have used riboprotein complexes, for example, using cationic lipids, and you can work with purified Cas9 and in vitro synthesized guide RNA to deliver. So again, this table is adapted from AdGene. If you're interested in some additional information about delivery, especially therapeutic delivery, this will be briefly discussed today in the Frontiers in Genome Editing talk. So assays to monitor editing. Shown here is an example of some indels that you can expect to generate using Cas9 targeted double-strand breaks. So very frequently, the indels that are created when targeting Cas9 are very small insertions or deletions, as shown here from some sequence clones. Larger insertions or deletions are possible, and they certainly do happen, but normally these small insertions or deletions are the most commonly observed. So there are two main ways that we are able to assay genome editing. And the first is called surveyor. And surveyor can be done in virtually any lab that has access to basic molecular biology equipment. And it can be done in a day, so it's pretty simple to use. The only main downfall with surveyor is that it's low throughput. To do surveyor, you first need to design primers spanning the target sites. And so if this is some of the pool of genomic DNA that you're working with, and there's an indel right here, you want to design primers so that there are about 500 to 800 base pairs of a PCR amplicon. You then want to amplify this genomic DNA. And it's very key. One of the key things about doing a surveyor protocol is to have a very clean amplicon, because any smears can alter your analysis. Next, you want to melt and re-anneal the DNA in some buffer. And so when you rehybridize, you go from this indel here, can rehybridize randomly with other genomic sequences. This creates bulges in the DNA. These bulges are recognized by the surveyor nuclease and cut, as shown here. You then run these fragments on a gel and can quantify using this formula. And here's an example of what the surveyor assay looks like. So uncut, so if you don't add surveyor nuclease, you see the single PCR amplicon. And then the cut fragments are shown here. And you can note that in this particular example, 280 and 270 add up to 650. And that's how you do surveyor. A higher throughput and especially these days recommended method to assay for on-target as well as off-target indels is by using next generation sequencing, or NGS. And to do that, you work through to do NGS, you PCR around your indel, and then you apply barcodes so that you know which region you're testing. You can pool and then sequence. And again, this is a great way to assay on-target and then predicted off-target regions. It also gives you sequence information to show you what the indels look like. If you're looking to really dig deep and figure out every single off-target cleavage event that occurs in your cell type of interest, you can also do these larger unbiased off-target detection methods. There are many different methods that have been described. This is a nice review that kind of describes all of them. These are usually quite laborious, unfortunately, but they're a really great way to get a very unbiased and whole genome-wide view of what cleavage events are occurring in the cell type of interest. Additionally, published very recently, are two other in vitro methods to detect, to do unbiased off-target detection. And these are circle-seq as well as site-seq. And so these might be additional methods that one could use to detect for off-target cleavage events within a cell. So lastly, before I end the talk today, I'm going to briefly elaborate on some additional applications of CRISPR-Cas tools, and then give a few anecdotes about doing HDR using CRISPR-Cas. So using HDR is it's a very common thing to do when working with Cas9. You're trying to knock in a particular mutation or a selectable marker, for example. And HDR, again, is a method to use to make precise genomic edits. To do HDR, all you need to do is to have a Cas nuclease cut very close to your target of interest, and then provide a template that you want to but provide a template to template the change that you're interested in making. So one of the key things that I'd like to highlight before I just go into a little bit more about doing HDR experiments is that CRISPR-Cas technologies generally work quite well in many different cell types. However, it's very key to note that HDR efficiency really depends on your cell type. And so you might have cutting events that occur, but HDR can often be the bottleneck when doing these experiments. And so I'll provide a few anecdotes on doing HDR, but if you're looking for particular information, I highly recommend that you attend these talks later during Genome Engineering 5.0. There's a nice talk about homology-independent targeted integration, and then there's another talk by David Liu about base editing. And so these might be of interest to you if you're looking to do HDR. To do HDR, again, you need to cut as close as possible to the site of interest. So this is actually done using talons, but can also be applied, this information can be applied using CRISPR-Cas tools. And it shows that the closer you are to cutting at the site that you're hoping to make your HDR change is ideal. In addition to cutting as close as possible to your HDR site, you need to provide a template, and templates can either be linearized plasmids or single-stranded oligonucleotides. Linearized plasmids are good if you're working with longer modifications, whereas SSODNs can be quite high efficiency, but they have very short lengths. A common question that people have is how long to make the homology arms. And so shown here are a few pieces of information, but ultimately it's good to research the literature and see what others have done. Short homology arms can be done. So some report a minimum of 80 base pairs in mammalian cells, whereas other papers have reported really long homology arms. So again, playing with this size, this is kind of a ballpark of the size of homology arms to work with when doing HDR. So again, Cas9 cleaves very efficiently, generally, in most cell types. And so to increase HDR efficiency, there are a few tricks and tools that people have developed. And one relates to the different ways that a double-strand break is repaired in a cell. So again, the predominant pathway for repairing a double-strand break is NHEJ. And so if you help inhibit that pathway, this can help promote the repair of a double-strand break using HDR. And so to suppress this pathway, you can use shRNA gene silencing to target some of these proteins, or there's also an SCR7 ligase 4 inhibitor that has been used. Another interesting method that has been described is shown here by Richardson et al. And it takes advantage of the mechanism by which Cas9 cuts its DNA. So when cutting the DNA, Cas9 is known to hold on to the strands of DNA shown here by these X's. Whereas this strand here is able to breathe, they say, or kind of comes out of this complex. And so after duplex cleavage, if there's a complementary DNA that anneals to the strand, it can help pull it away out of the site, leaving this area open and therefore more amenable for HDR. So a few other HDR strategies that I'll briefly mention One is by Paquette et al., and it's called correct. It's a two-step method for short HDR alterations that are distal to the PAM sequence, as shown here as M. So this method helps reduce a common problem that occurs because sometimes indels can occur in the same sequence as an HDR event, especially if it's a small alteration distal from the PAM. Another interesting method described here is an interesting co-selection strategy that allows you to enrich for cells that has undergone HDR. So in this method, you target your gene of interest as well as the gene ATPA1. Since HDR is often cell cycle dependent, if one gene undergoes HDR, it's likely that the other one may undergo HDR as well. And by targeting ATPA1, this generates a dominant gain-of-function allele that is resistant to a drug. And therefore, you can use this to select for cells that have undergone HDR. Besides using CRISPR-Cas tools to cut to create indels or to knock out a gene or to do HDR, CRISPR-Cas tools can be used in many other ways. For example, a very common way, or a very common thing to do with CRISPR-Cas tools is to use it as a screening approach. So the next talk is actually going to be on screening, so I will not elaborate further on that. CRISPR-Cas tools can also be used to control transcription, label DNA, and can also be inducibly utilized. So with that, we hope that CRISPR-Cas tools can be used for many different things, as shown in this nice schematic here. And yeah, so that's the end of my talk. This is the Feng Zhang lab. And with that, are there any questions? Well, if you don't have any questions now, feel free to come to any of us in the green t-shirts later during the talk.
Thanks for the talk. It was very informative. And you were showing the ways how to screen for the indels happening or for the editing happening. And you were showing the surveyor or the next generation sequencing. There is one method that is called TIDE. Have you heard about that? Did you check how efficiently it's detecting compared to surveyor or the next generation sequencing? Okay, we can discuss later. Yeah, thanks.
More questions?
Yeah, thanks for your nice talk. So my question is regarding the delivery.
Thanks.
So you mentioned a few delivery methods. So is there any way it's better than the others? Or the other question related to delivery is to say how effective are the methods, and then how do we care how much they get into the membrane and then get to the nucleus?
So are you asking
Three
which...
questions.
So, so which okay, so
Number one.
what's your first question? Yeah.
Different delivery methods. So from your experience, are there any ways one is better than the others?
Okay, so so preferred or suggested delivery
Sure,
methods.
sure.
So normally in our lab, we usually work with cell lines, and we work with a really easily transfectable hex cell line. And so we use lipofectamine, and we can get almost 95, almost very, very efficient transfection. So again, it'll really depend on which cell line you're working with. But if you're looking for a very easy-to-use cell line to simply just test to see if your guide works, hex cells are good to use. Although ideally, you want to do it in the cell type that you're interested in working with. If you're going to go in vivo, you also want to try to match a cell line that's similar in vitro than the cell line that you're going to work with in vivo. So it really just depends on your application.
Okay, thank you. The number two question is, say, how you monitor the efficiency, for example, into the cell membrane and then further into the nucleus.
So how do we monitor?
Or do we care how much get into the nucleus? So if you have the agent deliver into the membrane, right? Mm-hmm. What really function is they get into the nucleus? Is that right?
So are you asking, how do you monitor the efficiency of cleavage?
Yeah.
So if you're able to deliver the Cas9 plasmid into the cells, how do you then monitor to see how efficient Cas9 cleaves?
Sure, yes.
Okay, yeah. And so when doing that, we always recommend that you do a GFP control to see what your general transfection efficiency is. Okay. So let's say your transfection efficiency is 90%, so you get really good transfection. That can give you an idea of how many cells receive the plasmid of interest. And so if you're getting about 90% transfection, if you do your assays and you find that cutting efficiency is at 50%, that can kind of give you an idea that about half of your cells are cutting very nicely. But it usually is quite efficient. So.
Oh, okay, so the last one might sound kind of lame and stupid. We need to make sure they into the cell membrane and then go into the nucleus, right? Mm-hmm. Okay. So is there any way we monitor how much into the amount? So X amount go into the cell membrane, how much go into the nucleus? Is that important?
Oh, so that's a good question. So if you get the plasmid past the cell membrane into the cell, how do you know if it gets into the nucleus?
Yes, that's right.
Yes, that's a good question. So most of these plasmids that have Cas9 have protein little tags on them called NLS sequences, so nuclear localization sequences. And so they're engineered to enter the nucleus after the DNA gets into the cytoplasm, so that when the protein is translated, the NLS tag will recruit other proteins to bring it into the nucleus so that it can be active in the nucleus.
Okay, so the efficiency you mentioned earlier is actually the overall efficiency, go into the membrane and then go into the nucleus.
Yeah, exactly. Okay. So usually when you're testing for the efficiency of how well genome engineering works, you often look at the DNA. And looking at to see if there are indels at your target. And so since you're assaying the DNA, which is the final, so that is, to work on the DNA, the Cas9 enzyme needs to get into the nucleus. So that's already kind of accounting for that.
Thank you very much.
Sure.